Hi there. This is our lecture for microbial growth, for microbiology for health sciences. This corresponds to chapter 6 of your textbook. So we'll go ahead and get started. Today's roadmap is going to include things like growth requirements for life, especially prokaryotes. We're also going to talk about the formation of biofilms and their relevance to health professionals. Next, we're going to talk about the kinds of culture media as well as culturing methods. And then we're going to talk a little bit more about bacterial growth characteristics, how they divide, and some of the interesting things they can do with respect to endospores. Before we begin, I want to talk about two quick definitions because you're going to see these a few times throughout the lecture. These terms are usually paired with another descriptive word to help you understand what you're talking about. So if something is said to be obligate, that means that there's no choice. The organism requires it, it has to have it. If an organism is said to be facultative, that means that the organism is somewhat flexible and can adapt to changing conditions. So, as I mentioned in the roadmap, bacteria have certain requirements for growth just like we do. This is true across the living world. And so there are a couple terms I want to describe, and you'll see that there is a blank table over here, where's my mouse, on the bottom left. You're going to take a few minutes to fill this in. So psychrophiles, this word comes to us. Philes is going to mean loving, phobic, just like we saw with cells when we were talking about hydrophobic and hydrophilic things. Phobic things are afraid of. Philes or philic means loving. So in this case, psychrophiles means cold loving. And specifically organisms that are involved in food spoilage are typically called psychrotrophs. Trophs meaning something about their nutrition. Mesophiles, these are the ones that are usually most medically relevant. These are moderate temperature organisms, so they have a particular range that you're about to investigate. We also have organisms called thermophiles. This term means heat-loving. And then we have life at the extreme, usually in the archaea. These are super heat-loving organisms called the hyperthermophiles. So we have a table up here on the top left that is showing you some of the temperatures in context. For example, we have freezer temperature, and we have what the temperature at which water freezes, etc. And so between this information and the information that you can find starting on page 150 of your book, where it talks about these sorts of organisms, take a minute and fill this chart out with what the optimum growth temperature would be for this type of organism that's shown on the left, what the range is from such and such degree to such and such degree, and where you might find these organisms in the real world, or what their real world relevance is. So take a minute, push pause on me, and gather your resources and fill this chart out. Then resume the lecture. If you bring this chart to me filled in for class next time, it will be worth, there's one other chart that's coming, so if you fill out both of those, then that is worth 5 participation points for this lecture. Okay, so the temperature, what you should have found out when you were filling out your chart is that these organisms occupy a particular range on the temperature spectrum, where they have optimal growth conditions and where they could possibly grow. So their optimum is going to be depicted by these peaks that you see on these growth curves, or not really a growth curve, but where they are able to grow. So this is where they would love to be, but they will potentially grow anywhere underneath this curve. And so, for example, the thermophiles occupy a range of about 40 degrees to about 70 degrees. There's a little bit here where the mesophiles overlap with the thermophiles. And for this reason, you'll have certain organisms. We don't really occupy the same niche, so a lot of the times the thermophiles won't be important. But in this area, this is where you might see hot tub bacteria that can cause skin infections called folliculitis. The hyperthermophiles, you'll notice, they don't occupy the same temperature range as us at all. We're right around here at 37 degrees Celsius. So there's no way that we are going to occupy the same space as this. Okay. They're usually, like I said, ranges of about 30 degrees. If you look across, that's tends to be how they are. At the edges of those ranges, the organism can grow there, but it will grow at a much slower rate, and so that's kind of a problem. So, which of these, and maybe I've given it away by now, but which of these types of organisms do you think are the most medically relevant? It is the mesophiles, because look at where it's hovering over. Its peak is right around 36, 37 degrees Celsius. And boom, that's exactly where humans are. So these are the ones that are most relevant for us. Okay, another important physical requirement is pH. Most bacteria are what's considered neutrophiles. They'd like to hang out at a neutral pH of about 6.5 to 7.5. And for this reason, you'll notice that most acidic foods like sauerkraut and that sort of thing have very few food spoilage problems. We can have some acidophiles, so that means that organisms that love acid, these ones would grow in a pH of less than 5.5. It's depicted here with a purple curve. And one of the organisms that can do this is called lactobacillus. I've shown its range here in this turquoise color. This is a normal feature of the human vaginal microbiota. They also use it, a different species sometimes, to make yogurt. And then, of course, the archaea tend to live at the extremes, and so you will see some that live in this pH range of about 2.5 to 3.5. If we can have acidophiles, we can also have alkalophiles, which would mean base-loving organisms. These ones tend to like a slightly higher pH. Vibrio cholerae is one of these. This is the causative agent of cholera, or watery diarrhea. And you can see it can grow across a wide niche. It tends to prefer 8 pH, or a pH of 8, excuse me, and it's usually killed by the stomach acid. Stomach acid will be somewhere down in this range. But sometimes you're just the right place at the right time with a slightly higher pH in your stomach, perhaps, where the Vibrio is protected by a food particle, and then you could have this problem. Okay, so here's the second chart that you're going to want to take some time to fill out. But this concerns osmotic pressure. When we were talking about our cells and the importance of the cell wall for providing a counter pressure against an influx of water that would cause the cell to burst, that's kind of what we're describing here. Osmotic pressure, you can also kind of think of this as salinity or tonicity, and that's a term that's written down here on your chart. Microbes are about 80 to 90% water, a lot like we are, and they require nutrients from their surrounding medium. So we describe the liquid medium that they're surrounded in based on its tonicity. So you can have an isotonic solution, you can have a hypertonic solution, and you can have a hypotonic solution. So I want you to take a minute with this chart that you've printed out or recreated on your own, and you want to write how do the concentrations of solute on the outside versus the inside compare? Is it greater than, less than, or equal to? You're going to want to fill that out for all 3 conditions. Based on that, which way does the water move? Does it move from the outside of the cell into the cell? Does it move inside to out, or something else? And then what is the consequence for the cell? Is it going to lyse? Is it going to just hang out and be happy? I want you to take a few minutes and fill out this chart. Again, if you can bring to class these 2 completed charts, this one and the one on temperature range, that will be worth the 5 participation points that we lost on the snow day. So I'll give you a moment, push pause, and work on that, and then come back. Okay, so again, some more things on osmotic pressure. This has an enormous role in food preservation. We have these high salt, when you think about beef jerky, you basically are eating meat, and it wasn't really cooked so much as it was dehydrated. And it's dehydrated with air, but more specifically, it's dehydrated by exposing it or marinating it in a high salt solution. And that draws all the water out of the cells of the meat, and not just the meat, but any microbes that might be underlying it. That beef jerky at the end is so salty that there's no way potentially for a food spoilage organism to live there. We can have, we mentioned that we have to, for our cells, really kind of maintain a particular osmotic pressure to prevent lysis. But there are some organisms that really like high salt. In fact, we have organisms called the halophiles that love it. For an obligate halophile, so that means there's no choice, they have to have it, they require a high salt concentration of about 30%. These are called extreme halophiles. And one example is shown over here in this picture of a lake in the Ukraine. It's this red bacteria that is in this lake, and it's really pretty, actually. But yeah, they require that salt. And so if you tried to grow it in the laboratory, unless you were growing it in a media that was 30% salt, they wouldn't survive. We also have facultative halophiles that don't necessarily require high salt concentration, but they can tolerate it above the typical level. And then I said that for the acidophiles, of course, you can have alkalophiles. We don't really have that situation when it comes to salinity or osmotic pressure. We don't have any halophobic bacteria that we need to worry about. Everything seems to require at least a little solute in order to maintain an osmotic balance. So in case you were concerned about that last chart, here's a depiction graphically, here's my mouse, of what those different solutions that you were working on the previous slide would mean. So in an isotonic solution, there's no net movement whatsoever across the membrane because water is moving in at the same rate that it's moving out. And the reason for that is because the solute concentration is the same both on the inside and the outside of the cell. In a hypotonic solution, that means lower concentration. And so what that means is if we look at the cell, the concentration of solutes on the outside is much less than what's on the inside of the cell. So osmosis, in the process of osmosis, that water is going to move to try to equalize those two concentrations. And in so doing, it's going to overfill this cell. And much like a water balloon, it will burst eventually. The opposite situation, so this is called osmotic lysis. Another thing that can happen on the opposite angle is if the solution in the surrounding medium is higher for solutes than it is on the inside of the cell, then the water is going to rush out of the cell into the external environment to try to equalize those solute concentrations. And of course, this is a losing battle. There's no way that this tiny cell is going to be able to provide all the water that's needed here. But what happens is you'll see that the plasma membrane starts to withdraw and become detached from the cell wall. And this is called plasmolysis. Okay, so the organisms, in addition to needing certain temperatures and certain pH range and certain osmotic conditions, is also going to have certain chemical requirements. And these are the big four elements that most life requires, but especially our bacteria. If we were to grow a beaker or a flask of organism for a couple days, and then we spun it down and just isolated the bacteria and weighed it, if we were to get the dry cellular weight, 50% of that weight would be made up of carbon. And the reason why is because carbon is sort of the central element for all organic matter. So it's part of carbohydrates, it's part of proteins, it's part of nucleic acids, and within that is our ATP molecule, which is our energy, and also lipids. Second to that is nitrogen, which is present in about 14% of dry cellular weight. And it's a key component of proteins and nucleic acids. Sulfur is also an important molecule. And together with phosphorus, they make up about 4% of the estimated dry weight. For sulfur, this is an important molecule or element, for proteins. And phosphorus is important for both lipids. Remember the phospholipid bilayer of cells and how many how many of those phospholipids make up a plasma membrane. But they're also important for the construction of nucleic acids. Further, they also require trace elements, just like we do. At the bottom of your daily vitamin, you'll see that you're receiving a low concentration of things like magnesium and cobalt and zinc and other things. Microbes require very similar things. They require iron, copper, molybdenum, zinc, etc. These are usually considered, or within the cell, they are enzyme cofactors. So they're something that will bind an enzyme and help it to function. So, as you know, we require oxygen to live, and so do microbes. The reason we require oxygen is not just to inflate and deflate our lungs. Actually, your lungs are there to accept oxygen from the environment and deliver it to the bloodstream. Why is that important? Because every cell in your body has to have oxygen in order to harvest energy from glucose, which is the main fuel of the bloodstream. So, and cells. And the reason for that oxygen requirement is this thing right here. The very last step of or glucose metabolism, we go through glycolysis, we go through something called citric acid cycle, and we'll talk about this next lecture when we talk about metabolism too. But hopefully some of this is very familiar from other classes that you've taken. But at the very end of that is something called electron transport chain, which is a series of membrane-bound proteins in the plasma membrane for bacteria that are basically playing hot potato with highly charged electrons. And as they pass along to one another, eventually they are able to, what's going to happen is you're going to create water as well as ATP in the process. We'll get into some of the nitty-gritty of how that's done, but for now that's a simple enough explanation for the time being. So we have to have it. But it turns out that there are toxic byproducts of oxygen that can cause damage to cells. The normal oxygen that we use, or normal molecular oxygen that we use for this process that's depicted over here, and what's circulating through our body is O2. It's normal molecular oxygen. But we actually utilize toxic forms of oxygen in order to kill pathogens using our immune system. So here are some of the toxic byproducts of oxygen. I don't care so much if you know the chemical formula here. Just understand the name that's highlighted in purple and kind of what it's able to do. So in this case, singlet oxygen is actually normal oxygen just boosted to a higher energy state. This is an extremely reactive molecule that can damage nearby cells and cell components. Superoxide radicals also looks like regular O2 with a negative charge. It's a byproduct of normal cellular respiration where oxygen is used as the final electron acceptor. However, it's highly unstable and it tends to steal electrons from anything that's close by, including important biomolecules. Certain organisms, including ourselves, can overcome this using something called superoxide dismutase in order to neutralize that toxic version of oxygen. It effectively converts these along with two hydrogen ions into hydrogen peroxide and oxygen. Further, speaking of peroxide, there's the peroxide ion, or anion rather, which is also a toxic form of oxygen. This is part of hydrogen peroxide, and it has to be neutralized by the catalase enzyme. And in the process, will release bubbles. Or there's a second enzyme that you can use called peroxidase that will not produce those bubbles in the form of O2. But again, the idea is we're converting, in this case, hydrogen peroxide into water and oxygen. Lastly, there's another toxic form of oxygen called the hydroxyl radical. It's got this, if you look at the chemical formula, it's just OH with this tiny itty bitty dot. This is an intermediate form of oxygen and the most reactive of all. It's formed in the cytoplasm due to ionizing radiation. So that is kind of a problem. Okay, so the way we classify our organisms is based on their tolerance for oxygen, and the way they're able to adapt to certain oxygen conditions is contingent on whether they have the enzymes necessary to detoxify those toxic forms of oxygen that they're exposed to that we just talked about. So for aerobes, these are organisms that use oxygen as the final electron acceptor. You're going to need to get used to that term because that's how we define all of these things. It's not I need oxygen to breathe. They need oxygen as their final electron acceptor for energy harvest. And so they have enzymes such as catalase, superoxide dismutase, that allow toxic forms of oxygen to be neutralized. These would include organisms like the obligate aerobes, which are shown here on the left. We also have facultative aerobes that are ones that prefer oxygen. So if they were growing in a tube, you might see a big cluster of them up here at the surface where the oxygen concentration is greatest. But you might also see some turbidity lower in the tube because these are organisms that prefer oxygen but can harvest energy using something else, whether that's anaerobic respiration, where something other than oxygen is used as the final electron acceptor, or they can limp along with fermentation. And some examples of this include E. coli and many yeasts that we use for food production, actually. Lastly, on this slide, we have an organism called the microaerophile, or a group of organisms that are called microaerophiles. These are ones that do require oxygen for growth, but they need it in a very low concentration. So if we were looking at a tube, where's my mouse, of a growing culture of this organism, you wouldn't see the cluster of organisms at the top where the oxygen concentration is greatest. You also wouldn't see them clustered at the bottom where the concentration of oxygen might be the least. They're kind of in this Goldilocks zone, right in the middle, where they're getting just enough oxygen and they're sort of living right on that edge. And again, a lot of this has to do with the enzymes that they have to mitigate the damage of oxygen byproducts. For the anaerobes, the situation is a little bit different. They use something other than oxygen as the final electron acceptor in order to gain energy. Because oxygen is what's one of our most electronegative elements. It likes to steal electrons from other things. Because we're using something else, we're not getting quite as much energy from that electron transport chain. So we will have obligate anaerobes that have no choice. They're unable to use oxygen. It's toxic for them. In fact, these organisms, if you look on this chart here, lack the enzymes to neutralize the harmful forms of oxygen. So they can't tolerate it at all. So if we were growing a culture like this of an anaerobic organism, their turbidity would only be present in the bottom. The rest of this would be effectively clear because this is where the oxygen is located. So they would tend to drift to this concentration that's way down here where you can't see anything except for the organisms themselves. So examples of this would be the Clostridium genera. So this would include tetani and botulinum that cause tetanus and botulism respectively. These ones tend to form spores as well. And so usually if you step on a rusty nail, what you've just done is you've introduced an organism or a spore into a deep puncture wound would be kind of an anaerobic environment for them to develop their infection. Okay, you can also have what's called aerotolerant anaerobes. So these are organisms that can't use oxygen per se, but they're not necessarily going to be harmed by it either. So for example, they may have one enzyme like superoxide dismutase that allows partial neutralization of all these harmful forms of oxygen. So in that way, they can sort of tolerate it. And so if that were the case, you might see turbidity throughout the tube here because it really doesn't matter where the oxygen is, it's using something else. This would include things like the lactobacillus genera that we were talking about earlier for making yogurt, but also our lactose fermenting microbes that can make things like brewer's yeast or the organisms like brewer's yeast that can ferment lactose. Excuse me. Okay, we've talked about biofilms before, but now we're going to spend a little bit more time on it, going into a bit more depth. These, again, are thin, slimy communities of bacteria that are adhering to a surface. That is different from the kind of organism that we might normally think of as alone and unafraid swimming through a medium. Those kind of cells are called planktonic cells. That's highlighted here in purple. And so these organisms would swim through the medium and find a suitable place to attach. That could be respiratory epithelium, it could be a heart valve, it could be an indwelling medical device like a central line or a pacemaker or a hip replacement. But anyway, they'll see somewhere that they can attach and they'll go ahead and do so. And once they do land effectively, they'll form a thin monolayer of cells. My mouse is disappeared. I cannot see it. There it is. I was moving it too fast, I think. But they form this thin monolayer of cells, and then they start building scaffolding made out of a sugary substance called extracellular polymeric substance, or EPS. And this will support three-dimensional growth. Again, I can't find my mouse. There it is. So this growth, we're going to have it's going to look sort of like the skyscrapers of downtown Atlanta, where we'll have these towers of organisms with gaps in between, or channels in between for things like oxygen and water to pass through, along with nutrients and to carry away waste products too. Eventually, things might get a little bit crowded, and so the planktonic cells will break free from the biofilm pillars and go find a new surface to colonize. So that's how the three stages play out. We have attachment, followed by growth, followed by detachment and dispersal. So the way they're able to do this, how do we coordinate between cells that are unicellular, is to have this cell-to-cell communication using messenger molecules that are called autoinducers. Autoinducers stimulate gene expression in whatever cell receives the message. And those cells then amplify the message in addition to completing the indicated task that the autoinducer told it to do. So this results in a coordinated, functional, biological community where everybody's effectively speaking the same language. And it can be in a mixed community. It's not like Pseudomonas speaks one language of autoinducer and Vibrio doesn't understand what it's talking about. You can also have mixtures across the gram-positives and the gram-negatives sharing space. You can have a multitude of different kinds of bacteria in that biofilm. So once they make the autoinducer molecules, those will float around maybe in that extracellular polymeric substance and eventually make contact with the receptor molecule for that autoinducer on a neighboring cell. That cell will take in the signal, start to affect gene expression in here, and start to produce more autoinducers to pass or relay the message to others, but also other things. It may turn on virulence genes. It could turn on the genes for bioluminescence, which allows certain bacteria to glow in the dark. And other group behaviors like making a biofilm. So another thing that's cool about the biofilm from the organism's perspective is this is going to be, I call it friends with benefits. It provides the better ability to have bacterial sex with other cells. If you remember when we were talking about cells, you have the sex pilus that can extend from one organism that has these fimbrial proteins to a recipient cell that doesn't have that yet, and doesn't seem to have the sex pilus yet either. And so the plasmid, where did it go? Remember, we have that extra chromosomal piece of DNA, that circular piece of DNA that will code all sorts of things, but one of the things it codes for is this sex pilus, and that will transmit all the way through here to the recipient cell. And after this pilus is retracted, now this cell will start to express those genes, and it will start to look fuzzy on the outside like this one does. So the proximity of the organisms inside of that EPS makes this more likely. But that extra polymeric substance also is very protective. It's protective against desiccation or dehydration. Antibiotics have a hard time drifting through that substance to get to their targets. And so for that reason, biofilms tend to be about 1,000-fold more resistant to microbiocides than planktonic organisms. It's also protective against the human immune system. Our cells that are surveilling for foreign material, they can't get to their antigen, if they did know it was there, they may not be able to seek and destroy to get through there. Antibodies rather can't drift through there and make contact either. So in addition to the ability to have sex and share genetic material, those organisms are also able to share nutrients. So lots of benefits to the organisms to forming biofilms, which is why about 70% of human bacterial infections are estimated to be due to biofilms. There's a selective advantage to doing so. Again, these tend to be antibiotic-resistant infections. They can form on pretty much any indwelling medical device. This could include a central line, an intravenous catheter, a urinary catheter, titanium hip replacements, any sort of foreign body that we add in. we have certain organisms that do this really well. And they are like super biofilm formers. And this would include Pseudomonas as well as Candida, which is a yeast sort of infection. Keep in mind though, when you send clinical specimens to the lab from a patient that's infected with a biofilm, they are not actually examining bacteria in a biofilm form. They're examining them in their planktonic form. And what did we learn? Organisms that are in a biofilm tend to be 1,000-fold more resistant to any sort of antimicrobial. And so when we get an antibiogram report or the resistance or susceptibility profile from the lab, well, that's nice, but it's not very practical. It's not what's happening in vivo. Okay, so what are we doing to combat the scourge, so to speak, of biofilms? One thing that we're doing, since it's hard to treat the infection once it's already established, is to prevent that from happening in the first place by incorporating antimicrobials into surface materials. This will only work, of course, for abiotic substances. We can't exactly go in and powder coat all of our arteries and other body parts with antimicrobials. But for example, my dog's water bowl has this Microbial logo on it, and it's got this kind of really smooth surface, and the reason for that is it's embedded with antimicrobials to try to prevent attachment of biofilms. And so we're doing this across lots of different surfaces, not just dog bowls, but things that are medically relevant too. In the academic world, there's a lot of research into how can we disrupt those quorum sensing signals, those autoinducer molecules, so that the bacteria can't talk to one another. Finally, there is a human iron carrier that we have in our cells, and especially our lungs, called lactoferrin. And what it does is it finds it finds iron and it grabs it up. So it's not free floating in the environment. Well, it turns out that bacteria need iron. So if we're having this contest to try to find as much iron as possible, and humans have this molecule that'll do that for them, perhaps that can inhibit biofilm formation. So that's another area of research. We're going to switch gears a little bit and talk about culture media and methods, because this has a lot to do with bacterial growth too. And we're working with a lot of culture media in lab, not just this week, but in the weeks to come. So we want to define some of these terms. A culture medium is just any sort of nutrient material that favors the growth of microorganisms. Most of the time it's going to be in a liquid or an agar. Well, they're pretty much always going to be in a liquid or an agar form. So for liquid, it's going to be broth tubes or flasks. And when you see broth, it really is a lot like chicken broth or beef broth. Those are usually some of the big protein sources for this, and then we add other things to make it more nutritious. But culture medium is effectively chicken noodle soup for microbes. So with agar, though, we take that broth that we're working with and we add a semi-solid jello-like substance that we get from algae, and we pour all of this into a petri dish, and it hardens once it's cool. We can also pour it into test tubes, and so I want to talk about how this would appear. But once we do this, these are pretty heat stable once they're poured. I mean, they can maintain their shape and their integrity even in an incubator, where some substances might not. Okay, so let's see. Our agar plate is in a petri dish. It will solidify over time. A broth tube, because it's liquid, will tend to have this even surface at the top. You can also pour agar into a tube, and if the tube is completely upright, it will effectively look much the same, but more opaque because it's not a liquid, it's a solid. However, if we pour the agar into a tube that's at maybe an angle, we lay it on its side a bit and then pour it, gravity will help us out so that we get what's called an agar slant. And so here's what it looks like from the side. Here's what it would look like head on. And we use this for certain organisms in order to grow them and transport them. Any of our culture media, for the most part, are sold in powdered form, and you just reconstitute it in water and then sterilize it. So we have some more definitions. Uh, when we're transferring something to this media that we're talking about, that's called an inoculum. This is the clump of microbes that are used to initiate that new growth. To inoculate something is the act of transferring microbes to that new environment. A culture is the microbes that grow from that inoculum that we started out with. And a third definition that's here is sterile or sterility. This is the idea that in that media, there are no living things present. This is very important for culturing. Remember that contamination is the enemy, but we sterilize all of our media before we work with it because we don't want to be confused later on, and we also don't want the inoculum that we're putting inside of that media to be having to compete with some underlying contaminant for resources and other things, because that's what we want to grow, not whatever might be in the background. So by sterilizing our media, this is how we trust that what we put in the medium will be the only thing that grows. We have several different kinds of media that we can work with. For the most part, so far, we have been working with complex media, which is called, or it's more of like a general purpose, anything could possibly grow here. This will include extracts of yeast or meat, but the composition can vary from batch to batch. Like if I make it one day, maybe on this day, the contents of the powdered form may have settled, and so maybe there's more salt at the top end of the bottle. With chemically defined media, the exact quantities of ingredients is standardized. So no matter which batch you make, we know exactly how much sodium is there, how much magnesium is there, et cetera, et cetera. Some organisms need a lot of growth factors. These are a bit harder to grow, and so that term is called fastidious organisms. So things that, they're kind of high maintenance organisms. We also have what's called reducing media. These will reduce the oxygen that's dissolved in the media to help an obligate anaerobic bacteria to grow. So these ingredients have been chemically modified to do that. And this media will often be used in combination with an anaerobe jar to further limit the amount of oxygen that's present. Finally, if we're growing something like, let's say we have a patient and we suspect that they may have an E. coli 0157H7 infection. They're having bloody diarrhea, et cetera, et cetera. Well, the problem is, that's kind of like trying to find a needle in the haystack of the colon, because your colon at any given time usually has about 5 pounds worth of normal flora E. coli in there too. So how do we separate the bad guy E. coli from the typical friendly neighbor E. coli? And so we might use something that's like an enrichment culture, where over time we're able to pick apart the true enemy in this case, or to find sort of those organisms that are present in low numbers. So I mentioned the anaerobic growth chambers, the jars, but so here's what that would look like. There's a jar, you put your plates inside of it, and these are you put this packet in here that's kind of one of those, if you've ever used those hot hands or a Thermacare heat wrap, once you open the package and it's exposed to oxygen, it carries out a chemical reaction. So that is happening here too. You have this envelope that you open, and it starts to exchange oxygen for CO2. And you'll have an indicator strip in here also that will confirm to you that that has taken place. You close the jar and you seal it shut, and you would then put this in the incubator. On the flip side, you can also have these anaerobic chambers to work with, where you would put your plates in here in the airlock, close the door, and then you put your arms inside of these arm ports, which it's kind of like working in a NICU with babies. It's a similar kind of idea, I guess. But in this case, it's a very low O2 environment in here. And so you would put your arms in, and then you could streak, you could do other things with your anaerobes. So some more different kinds of media include the selective media. These are ones that contain dyes or antibiotic that will inhibit the growth of certain organisms. Like maybe we only want to grow gram negatives, or maybe we only want to grow gram positives. There are ways to do that based on what's embedded in the media. We could also have differential media, which uses certain chemicals or nutrients inside of the agar or the broth, like pH indicators that will allow you to pick out a desired organism from a mixed population. Like, let's say we want to know, or we know that our organism is a lactose fermenter. So as far as finding the needle in the haystack, we don't want to look at all the bacteria. Just show me all of the lactose fermenting colonies, and then we'll figure out what we're working with. But often you'll be working with, you might be working with a pure culture by this time, or a nearly pure culture. So you can also combine these two things. You can have selective and differential media. And this is one example here is the McConkey agar that you're looking at right here on the right hand side, or actually this entire thing is McConkey agar. And so this will only allow the growth of gram-negative bacteria, and we can further distinguish the organisms based on their lactose fermentation. So if the organism is fermenting lactose, the colonies will be pink. If they are not lactose fermenters, then the plate will be sort of this colorless agar color sort of thing. You can review table 6.5 in your book to go over the different types of media and to kind of quiz yourself over time. Another kind of differential media is blood agar, and this it's differential because it has blood in it, usually from sheep or other sources, and certain organisms can do what's called beta hemolysis, where they will lyse red blood cells. They will lyse red blood cells, and on a blood agar plate, what that will look like is you'll have this clearing zone around your colonies. There's also a selective and differential media called mannitol salt agar that we're using here to grow Staphylococcus. You'll see if we look here at this third of the plate, this is an uninoculated spot. Here's what Staph epidermidis looks like, and this is Staphylococcus aureus. So the color change is based on the ability, to ferment mannitol. So this right here looks like Staphylococcus aureus is doing that. This is a high salt media, and that is what allows only Staphylococcus to grow on this media. So that's nice. Okay, sort of an odds and ends sort of thing. We do have some organisms that can only grow in living host cells. And your clinical lab can do this using cell culture. Viruses are especially one of the things that this will apply to, but we have certain bacteria that require cell culture growth as well. And so this requires a lot of special handling. Another important or sort of oddity is that certain organisms need slightly higher carbon dioxide concentrations than what's available in the atmosphere. So we have special CO2 incubators where that is elevated slightly. And the organisms that prefer that, we call those capnophiles. So interesting. Another interesting thing having to do with how we grow organisms and how we manipulate organisms is that not all flavors of bacteria are the same. And some of them are more pathogenic than others or easier to transmit than others. So for that reason, many public health entities across the world have come up with this biosafety level strategy for growing things and working with them. So what we work with here at MGA is mostly BSL-1. I say mostly because we do work with one BSL-2 pathogen right now. But it's BSL-1s are ones that don't really need containment. We know a lot about them, and they're unlikely to cause disease in healthy individuals. This would include things like E. coli, and they really don't present a big threat to the people that are working with them or the environment if they happen to get loose. For a BSL-2, I mentioned that we do work with one organism that falls into this category, and that's Staphylococcus aureus. It didn't used to be a BSL-2, but with the advent of MRSA, and especially community-acquired MRSA, it is taking on a bit more prominence and risk, and so it is now defined as a BSL-2, and we are sort of adapting our laboratories for that. So we have, in this case, now we do have to contain them. We have to have, somewhat restricted access into the laboratory. We use keys, points of contact, that sort of thing. There is a moderate risk of exposure, especially if you mishandle the organism. But the disease sort of varies on where it was inoculated, that sort of thing too. Other organisms or pathogens that fall into this category include influenza, HIV, as well as Lyme disease. For BSL-3, these are organisms they can be either something that is normally found in our environment or something that we've sort of imported from elsewhere, like an exotic agent, that can cause disease. They tend to be transmitted by aerosol transmission or in the air, and they can cause serious or potentially lethal disease. So these require high containment. And this would include tuberculosis. Finally, the one that usually gets the most airplay or the most notoriety in Hollywood or in books is the BSL-4 laboratory organisms. And so this is what you would consider max containment, or like a maximum security prison for microbes. But again, these are specifically going to be the things that are high risk agents. Often they're considered like the exotic pathogens. And they are life-threatening. The most prominent example, especially with recent things where we sort of imported Ebola into the United States inadvertently, is the Ebola virus. These are dangerous, and so for that reason, there's a lot more security and a lot more personal protective gear, as well as infrastructure of the laboratory to protect personnel and the people around them. So in a BSL-3, you'll notice he's wearing a suit here, a pretty flexible suit. It's pretty thin. He does have a dedicated air source that is filtering into his cap, and there's a mask that's around his head. You'll see that his ear and his neck is still exposed, but he's able to work and relative comfort. BSL-4s are a little bit more advanced. These guys, they're wearing like the full Gumby sort of suit. So they're wearing something like this on the inside, but then also wearing sort of this rubbery suit on the outside as well. They have their own dedicated hoses for air. And then this is quite an ordeal to be doing this. To get into this, first of all, there's serious access control, usually like a fingerprint and a retina scan in order to get into the laboratory. You put on scrubs. You put on a full protective bodysuit, and then you can go work in the lab. And usually once you start work in the lab, you try not to leave until you have to, because it's quite an ordeal to leave, even to use the restroom. So they'll come out, they'll have to take a decontamination shower while wearing this suit. In case any aerosols have landed on the suit, the water will hopefully wash that away. Then they take off the full body protective suit and take another shower, this time of their person. And once that's complete, they can put their street clothes on and go back home. And we don't have a lot of those in the United States, but we have enough. We have enough for the time being. All right. So let's talk a little bit more about bacterial growth. When we talk about growth of an organism, generally, you probably are thinking like, for humans, that would be going from a fetus to a newborn to an infant, to a child, to a teenager, to an adult. And in that way, the organism itself is getting bigger. With bacteria, they're unicellular organisms, right? So in this case, we're not necessarily talking about the increase in cell size. We're talking about the increase in total bacterial cell numbers. So just understand that distinction. Bacteria divide by a process called binary fission. There's a diagram of it over here. When this happens, you have the bacterial chromosome. It's duplicated, and it will sort of anchor into little spots here in the plasma membrane and cell wall. You'll start to get this pinching in. It's not truly pinching in. What's actually happening is plasma membrane is being built in this intervening space, and so it's starting to. It looks like it's pinching inward, and that plasma membrane is being built right here along what's called like the septum, trying to meet the other side. Once it does go ahead and meet that other side, these can now pinch off from one another and be independent cells. Notice that we go from one cell to two cells here. And so that's a phenomenon you're going to see no matter what. In this case, we're going to go from one cell to two over time. And the amount of time that it takes to go from, let me back up first 1 second, to go from this step in the process to this step in the process, a full cell division, is called the generation time. So the generation time is really the time span between generations, but within bacteria we say between cell divisions. For us, our generation time is the time from which you're born until the time that you're old enough to reproduce. And so usually that's like 13, 14, 15 years in our case, at a minimum. But our fertility rate tends to be a little bit later in life. For E. coli, their generation time is about 20 minutes long. And they will go from one cell to two, from two cells. I want you to understand that they'll go, it's an exponential growth, because we're going to go from one cell, where's my pointer, one cell to two cells. Those two cells will divide into 4 cells and into the 8 cells, and so on and so on. So we're not going linear. We're not going 1, 2, 3, 4, or 5 cells. We're doubling every time. So E. coli has a relatively short generation time. It's 20 minutes long. Mycobacterium tuberculosis has a longer one, and syphilis is very long. And so for some of these organisms, they just grow so slowly that we've actually started to move away from culture as a means of detection and diagnosis. We've started working directly with DNA from a clinical specimen, because that's present in much smaller quantities, and we're able to amplify that and manipulate it easier than we are the culture, and get a faster answer for the patient, which is really important. So each time, I think I've already highlighted this, that every time the cells divide, the population doubles, and because of that we get exponential growth. So the way this plays out, let's say we take an inoculum of bacteria, a colony from a plate, and we introduce it to broth. Initially, they're going to go through what's called a lag phase. What we're looking at here is a time course on the bottom with respect to the number of bacterial cells that are present. And we measure this usually by the turbidity or the cloudiness of the solution. Because as bacteria grow, they tend to turn the broth solution into sort of a cloudy appearance. So initially they're gonna arrive at their new media, and they're gonna try to take stock of where they are. What kind of nutrients are available, what oxygen levels are present, that sort of thing. And this is called the lag phase, as they're adjusting for population growth, but there's no true increase in the population itself. Once they figure out what they're doing, they'll start to divide, and they'll divide at an exponential rate. This is called the logarithmic or log phase. And this is, the bacteria during this time are doing that binary fission. Our yeast are doing mitosis, just like our cells do. Eventually, though, they're going to start to run out of nutrients, and they're also going to accumulate toxic waste products. Between these two things, this is going to start to cause cell death. And during the stationary phase, you're going to have the same number of deaths as you see new cells or cell divisions. And so for that reason, we see this plateau. After that, you have what's called the death phase, where the cells are mostly dying now. They're not dividing anymore, and they will decrease at a logarithmic rate as well. This is a typical growth curve for the Staphylococcus species, or genera, but you'll see this phenomenon across many organisms. One sec. Okay. One kind of cool thing that bacteria can do, though, with respect to growth, is form endospores. Now, this was something that was in our cells chapter of our book, but we skipped it during cells because I didn't think it made a lot of sense to talk about it until we talked about growth. Because these organisms that can form spores, they're gram-positives, they can build little lifeboats that we call endospores. And what these are really durable dehydrated cells, with thickened cell walls and extra layers to protect them. When they're released from an organism, they are resistant to heat, they're resistant to drought, toxic chemicals, and radiation. So here's an electron micrograph of a Bacillus subtilis that can do this. So this right out here is probably the vegetative cell, or what would normally be there, and this is the endospore that's on the inside. So I mentioned that the ordinary cell is called a vegetative cell. That doesn't mean like we think of it in the human situation, where when people go into a vegetative state, that they're no longer cognizant of what's going on. These are normally living cells. And this cell will sense that a key nutrient is becoming scarce or unavailable. So it will start to trigger this process called sporulation that can take several hours. What happens is first the chromosome is going to be duplicated and it's going to migrate over to this one spot. You remember how in binary fission we would see that plasma membrane starting to be synthesized into the interior environment. These organisms will not just start to build that plasma membrane, a little bit at least, but they'll also start to pull this membrane away from the cell wall. Sorry, this purple structure here is cell wall. The yellow structure is plasma membrane. And so this plasma membrane is starting to pinch inward and be synthesized inward. Eventually you'll have this zone of clearing in between that is the plasma membrane separating this cytoplasm from the rest of the cytoplasm. And now we'll have this entire septum surrounding this isolated portion. This is called the forespore. Next, you're going to have a phospholipid bilayer or cell wall layer that will form between these two membranes that have now formed. A spore coat is going to be added, this purpley pinkish structure on the outside. And now the cell is going to start to lyse. You'll notice that the original cell's DNA is being degraded, most likely because of harmful conditions in the environment that are now taking its toll on the organism. But the lifeboat will survive to live another day. These spores, when they're formed like this, they are not biologically active. They are not carrying out metabolic reactions. They are effectively like a time capsule. There's no enzymatic activity at all. And they can remain dormant for thousands of years. We can isolate endospore from things like permafrost that are millions of years old. And when we grow them in the laboratory, or we expose them to the right conditions in the laboratory, they perk right back up. Let's talk about how that happens. That's a process. If we sporulate in order to make the spore, to hatch that spore, so to speak, is called germination. This is when an endospore returns to the vegetative state. And this can be triggered by several things. One is high heat, the presence of alanine, an amino acid that's present in many proteins. That would be a good indicator that it's a nutrient-rich environment. And also a derivative of the nucleotides that you might be more familiar with that's called inosine. When the endospore is able to sense this in the environment, the endospore enzymes that have been dormant this whole time will start to digest the outer layers of the endospore, that coat area, the outer membrane, the cell wall, and then finally the plasma membrane, ever so slightly, in order to allow water into the endospore. Once that happens, full metabolism resumes and the cell becomes a vegetative, or the endospore becomes a vegetative cell once again. In this transaction, so to speak, we start with one cell and we end with one cell. So this isn't binary fission or true reproduction, because if that were the case, we would go from one cell to two daughter cells. But it's just a one-for-one replacement. This is an item of special concern in the food industry because we try to preserve foods often with high heat, especially in canning. But when you do so, you might be inadvertently activating or germinating spores that can cause foodborne illness, including Bacillus and Clostridium. Both of those are endospore formers. So there are some review slides for you to consider. This one is taken directly from your book. I would encourage you to work through the questions. We will be having an exam soon with a review session. So work on these problems, and anything that you're having a hard time with, you can come to that review session and we can work through those things together. So there will be more on that in the laboratory or in class and laboratory soon. But thank you for watching, and I'll see you next week.